Welcome back to Anatomiqa. In this session, we will introduce ourselves to embryology and try to understand what is embryology. The learning objectives of the session are: you should be able to describe the stages of human life and explain the terms phylogeny, ontogeny, trimester, and viability. What is embryology? How do we define embryology? It is the science that deals with processes and regulations in the prenatal growth and development of an organism or the individual in the female genital tract. It includes the study of gametes, their fusion to form zygote, and the further development up to the birth of the fetus. It is also called developmental anatomy. Prenatal development involves repeated division of most of the cells in the body, resulting in increase in size, complexity, structural and functional differentiation of the body. Embryology includes the study of astonishing integration of various complex molecular, cellular and structural processes. These are accountable for the growth and development of a 9-month-old neonate containing, guess how many cells are present in the neonate? It is 5 to 7 into 10 to the power of 12 cells, which is formed from a single-celled zygote. Do you have any idea when and where does the development of an individual start? It all starts from a very specialized cells called primordial germ cells. Primordial germ cells can be first identified within the wall of the yolk sac during the 4th to 6th weeks of gestation. Yolk sac is one of the extraembryonic membranes that nourishes the embryo in early stage of development. These primordial germ cells will give rise to the germ line, a series of cells that form the sex cells or gametes, which is the ova in the female ovary and sperms in the male testis. However, these gametes will not function to form the next generation for several decades until the onset of puberty. Yet, remarkably, one of the first things that happens in the developing embryo is to set aside the germline for the next generation or the future generation. Similarly, the germ lines that gave rise to the developing embryo were established a generation earlier when the embryo's father and mother were developing in utero, that is, when the embryo's maternal and paternal grandmothers were pregnant with the embryo's father and mother. Is this clear? Let me make it more clear. Let us start the journey of your development. Imagine your father and mother as embryos developing in their mother's womb. The germ cells needed for your development is formed in your father's and mother's embryos when they were aged about 4 to 5 weeks in the womb of your paternal grandmother and maternal grandmother respectively when they were pregnant. Hence, your development started in your paternal and maternal grandmother's womb. To go back and check the history of embryology, according to Aristotle, recognized as the first embryologist in history, the pursuit of science begins with a sense of wonder. He posited that people engage in philosophy because of this wonder, which serves as the starting point for acquiring knowledge. Throughout history, the process of an animal developing from an egg has consistently evoked a sense of wonder. For example, the act of delicately cracking open a chick egg each day during its 3-week incubation reveals a captivating transformation where A thin band of cells give rise to an entire bird. Aristotle conducted this procedure, observing the emergence of major organs. For a layman, this is amazing and remarkable phenomenon, but for a scientist, he wants to uncover the mechanisms underlying development. Rather than diminishing wonder, gaining new insights into the process only deepens our appreciation. Now, when we see multicellular organisms, they don't instantaneously appear in their final forms. Instead, their existence unfolds through a gradual process of beautiful journey with progressive changes known as development. In nearly all instances, the journey of a multicellular organism begins with a single cell, the fertilized egg or zygote, which undergoes mitotic division to generate all the cells constituting the body. The study of animal development has traditionally been referred to as embryology, focusing on the stage between fertilization and the birth. However, development extends beyond birth and even adulthood, as most organisms continue to develop throughout their lives. Certain animals can regenerate their lost body parts, and many species undergo metamorphosis, such as the transformation of a tadpole into a frog or a caterpillar into a butterfly. On a daily basis, as humans, we replace over a gram of skin cells, shedding older cells as we move, and our bone marrow continually produces millions of new red blood cells every minute. Consequently, modern terminology leans towards describing developmental biology as one of the major disciplines that investigates embryonic and other developmental processes. As we become multicellular organisms, development accomplishes two major objectives. It generates cellular diversity and order within each generation, and it ensures the continuity of life from one generation to the next. Thus, there are two fundamental questions in the developmental biology. How does the fertilized egg give rise to the adult body, and how does that adult body produce yet another body? These two huge questions have been subdivided into 6 general questions scrutinized by developmental biologists. The question of differentiation. A single cell, the fertilized egg, gives rise to hundreds of different cell types: muscle cells, epidermal cells, neurons, lens cells, lymphocytes, blood cells, fat cells, and so on. This generation of cellular diversity is called differentiation. Since each cell of the body, with very few exceptions, contains the same set of genes, we need to understand how this same set of genetic instructions can produce different types of cells. Now the question is, how can the fertilized egg generate so many different cell types? The second question of morphogenesis. Our differentiated cells are not randomly distributed. Rather, they are organized into intricate tissues and organs. These organs are arranged in a way there is same type of orderly arrangement each one of us. The muscles, same shape and numbers in each region or part of the body. For example, biceps brachii is spindle-shaped in all individuals. It has 2 heads in all the individuals. Fingers are always at the tips of our hands, never in the middle or in any part of the body. The ears are always in our heads, not in our toes or gut. This creation of ordered form is called morphogenesis. Now the question arises, How can the cells form such ordered structures? The question of growth. How do our cells know when to stop dividing? If each cell in our body were to undergo just one more cell division more than required, it would lead to horrible malformation, isn't it? For example, if each cell in our limbs underwent just one more round of cell division, we could tie our shoelaces without even bending our body. The face, trunk, limbs, everything is proportional to each other. Now, what will be the question? How is cell division so tightly regulated? The question of reproduction. The sperm and egg are very specialized cells. Only they can transmit the instructions for making an organism from one generation to the next. How are these cells set apart to form the next generation? And what are the instructions in the nucleus and cytoplasm that allow them to function this way? The next thing is the question of evolution. Evolution involves inherited changes in the development. When we say that today's human being is erect and called Homo sapiens, we are saying that changes in the development of cartilage and muscles occurred over many generations in the embryos of the human ancestors. The modern scientific evidence shows that we have evolved from monkeys. In Indian culture, we respect monkey, do puja and pray. This message was given to us thousands of years back. Our ancestors knew our evolution. Recall Dashavatara, Matsya, the fish, that is purely aquatic animal. Kurma, which is the turtle, adapted for both water and land. Varaha, the wild boar, completely adapted as land animal. Narasimha, representing the evolution of man from animal. Vamana, Parashurama, Rama, Krishna, Balarama or Buddha, representing the evolution of man from Australopithecus, the dwarf and bent, to the modern Homo sapiens, the erect and intelligent man. The 10th form, Kalki, which still has not happened, may be the evolution of AI, man, which we do not know. Can be anything. Now we have come to an era where AI is ruling us. So the question is, how do changes in the development create new body forms? Which heritable changes are possible given the constraints imposed by the necessity of the organism to survive as it develops? Now comes the question of environmental integration. The development of many organisms is influenced by the environment. You would have observed people always being angry or short-tempered, some always, no matter what, are joyful, and some always depressed or worried for every small thing. The people who are sad, depressed, angry can take the help of the tools like yoga, meditation, or physical exercise, which can help people overcome their behavior and become joyful in their life. These tools are not restricted only to help behavior. It can be applied to diseases like diabetes, hypertension, cancer, etc. The change in right type of food and lifestyle of the individual can free them from the disease. The behavior of the human depends on the environment each individual is brought up and it can be changed. So the question now is, how is the development of an organism integrated into the larger context of its habitat? All these questions can be answered through the research and experiments and have been answered. Okay, for some questions, answers are discovered, but for many, it is still yet to be discovered. Now coming to the subdivisions of embryology. Embryology itself is a part of anatomy, and now we will check what are the different subdivisions of embryology. It can be studied under the following subdivisions. General embryology, it is the study of development during pre-embryonic period, and embryonic period, which is the first 8 weeks after fertilization. During this period, the single cell zygote is converted by cell multiplication, migration, and reorganization into a miniature form of an individual with various organs and organ systems of the body. Systemic embryology, it is the detailed study of formation of primordia and their structural and early functional organization into various organs and systems of the body. It is further subdivided into development of cardiovascular system, digestive system, urinary system, genital system, etc. Here you can observe the heart beating, which starts as early as the 4th to 6th week of gestation. Comparative embryology, it is the study of embryos in different species of animals. Experimental embryology, it is for understanding the effects of certain drugs, environmental changes that are induced like exposure to radiation, stress on the growth and development of embryos and fetuses of lower animals. The knowledge gained from these experiments can be used for avoiding the harmful effects of these factors in the human development. It is a vigorous and promising branch of embryology. Biochemical and molecular aspects in embryology, such as chromosomes, gene sequencing, and regulation, play an important role in the development of embryo. Teratology. This is a branch of embryology that deals with abnormal embryonic and fetal development, that is, congenital abnormalities or birth defects. Now coming to the importance of embryology in the medical profession. It bridges the gap between prenatal development and obstetrics, pediatrics, perinatal medicines, and clinical anatomy. It is of practical value in helping to understand the cause of variations in human structures. It highlights gross anatomy and explains how normal and abnormal relations of structures develop. Embryology can be helpful in healthcare strategies for better reproductive outcome. Knowledge of embryology facilitates interpretation of the results of various techniques such as fetal ultrasound, amniocentesis, and chorionic villus biopsy. Based on the results, appropriate treatment can be planned. Example, prenatal surgeries are performed for correction of a defect in the diaphragm, postnatal correction of a cardiac defect, medical line of management of a diabetic or hypertensive mother. Therapeutic procedures for infertility, fertility-related problems. If the woman is unable to conceive by natural methods, alternate methods like cloning, which is developing another individual of tissue or organ from the somatic cells, and in vitro fertilization, which is fertilization outside the individual's body, can be planned. Here you can see the artificially induced sperm into the ovum. For preventing the pregnancies, various birth control methods, both medical like contraceptive pills and surgical techniques such as vasectomy and tubectomy are available. A basic knowledge of embryology is required for understanding the mechanism of action of these methods. Stem cell therapy. Undifferentiated cells forming tissues in the embryo are called stem cells. These undifferentiated cells can differentiate into various specialized cell types depending on the signal it receives. These cells will be capable of treating certain diseases in postnatal life. Let us go through some of the terminologies and basic processes in embryology. Growth differentiation are the two basic processes involved in the conversion of a single-celled zygote into a multicellular human newborn. Organizer. Any part of the embryo which exerts a morphogenetic stimulus on an adjacent part or parts is an organizer. There are three types of organizers. An organizer is formally defined as a region or group of cells in an embryo that can both induce and pattern adjacent embryonic cells. Inducers are that group of cells which can change the fate of another group of cells to develop into particular organ or tissue, and pattern is that which generates an organized set of structures. To date, about 4 such regions have been demonstrated. The primary or Spearman organizer, also called Ensign's node in amniotes. The notochord, the zone of polarizing activity of the limb bud, the mid and hindbrain boundary. Stem cells. These are undifferentiated cells having capacity to differentiate into various types of cells by replication. Reproduction. It is a mechanism to produce new generations of the species continuously. For reproduction, vertebrates require the presence of two different sexes, that is male and female, which differ in external and internal sex characters. The internal sex organs, that is gonads, testis and ovary, produce gametes, sperm and ova, that differ in each sex. Note the difference between gonads and gametes. Gonads are the paired sex glands, testis in male and ovary in female. They are responsible for the production of gametes or sex cells, that is sperms and ova, from the primordial germ cells that carry out the special function of reproduction. The process of formation of spermatozoa is called spermatogenesis, while the process of formation of ova is called oogenesis. Spermatogenesis occurs in the testis, while oogenesis occurs in ovaries. The two are collectively referred to as gametogenesis. Fertilization is the development of a new individual which begins at the moment when one male gamete, that is sperm or spermatozoon, meets and fuses with one female gamete, that is ovum or oocyte. The process of fusion of male and female gametes, which includes several steps, is called fertilization. Fusion of male and female gametes results in the formation of a single-celled structure called zygote. The zygote multiplies and reorganizes to form the miniature of new individual called embryo that grows and matures as fetus in the mother's womb and delivered at the end of term pregnancy. With these terminologies, let us now define what is the development of a human being. Development is defined as the process where someone or something grows or changes and becomes more advanced. Human development is a continuous process that does not stop at birth. It continues even after birth for increase in the size of the body. Development is divided into phases. Development occurs both before birth and after birth. Development before birth is called prenatal development. Development after birth is called postnatal development. Each is further subdivided into several stages. Prenatal development is the process of progressing from a single cell through the period of establishing organ primordia in the first 8 weeks of human development and the period from that point on until birth, a time when differentiation continues while the fetus grows and gains weight. There are 3 stages in prenatal development: pre-implantation or pre-embryonic period, embryonic period, and fetal period. Pre-implantation period spans from fertilization to end of second week of gestation. It extends from fusion of male and female gametes to form single-celled zygote to formation of primitive germ cell layers of developing organism. It includes first and second weeks of intrauterine development. The following morphogenetic events takes place during this period: fertilization, fusion of male and female gametes resulting in the formation of zygote; cleavage, a series of mitotic divisions of zygote resulting in the formation of morula; morula transportation towards the uterus. That is, morula, as it is forming, it passes through the uterine tube towards the uterine cavity. Formation of blastocyst with 2 groups of cells, embryoblast and trophoblast. Embryoblast differentiates into primitive 2-layered germ disc called bilaminar germ disc. Trophoblast differentiates into cytotrophoblast and syncytiotrophoblast. And initiation of implantation into the wall of the uterus. These are the changes occurring during the first part of the embryonic period. Embryonic period spans from 3rd week to 8th week of gestation. The following morphogenetic events take place during this period: differentiation of trilaminar germ disc, that is, formation of 3-layered germ disc with the appearance of mesoderm in between ectoderm and endoderm; early organogenesis, formation of primordia of various organs such as lungs, heart, liver, etc.; formation of extra embryonic supportive organs and membranes, placenta, umbilical cord, and amnion or amniotic membrane. Fetal period extends from 9th week to 9th month. This includes the following: growth of fetus in all dimensions and specialization of various body structures. With this, now let us see the postnatal period of development. It extends from birth of an individual to adulthood. The various stages in postnatal development are as follows: neonatal period, it extends from birth to first 28 days after delivery. These first 4 weeks are critical in the life of the newborn or neonate, as various systems, especially respiratory and cardiovascular, has to make adjustments with the external or extrauterine environment. The branch of medicine that takes care of neonates is called neonatology. The branch of medicine that take care of the fetus from 28th week of intrauterine life and newborn up to 6th day of extrauterine life is called perinatology. Infancy, it extends from 1 month to 1 year, and the newborn during this period is called infant. Childhood, it extends from 2nd year to 12th year of age, and an individual is called a child. It is the period of rapid growth and development. This age is also called pediatric age. We all are well known that the medical branch that deals with the infants and children is called pediatrics, and the specialist who treats them is called pediatrician. Puberty, it extends from 12 years to 16 years. there will be rapid physical growth and development of secondary sex characters that depend on the interaction of sex hormones and growth hormones. Adolescence, it extends from 17 years to 20 years. During this period, there will be rapid physical growth and sexual maturation. The reproductive ability is established. Adulthood, it extends from 21 years to 40 years. Middle age, it extends from 40 years to 60 years. And old age extends from more than 60 years to death. Now we have gone through different stages of prenatal and postnatal development of an individual. The complete life cycle of an organism involving these two stages, prenatal and postnatal periods of development, is called ontogeny. It is the expression of blueprint of life hidden in genes. It includes progressive changes followed by retrogressive changes. It involves various processes such as cell division, differentiation, and growth. Phylogeny, it is the evolutionary or ancestral history of a group of organism or species. It includes developmental changes in various organs like kidney or heart and organ systems like respiratory or skeletal systems, starting from fishes, amphibians, reptiles, birds, and mammals. So, when we say ontogeny repeats phylogeny, it is the life cycle of an organism which repeats its ancestral history. This is observed in the development of certain organs, which is retained in the human development, namely heart, lung, and kidney. To continue, we will see what is pregnancy or gestation. We have discussed prenatal development and its phases. All these are happening in the mother's womb. This process or period of development during which one or more offspring develop inside the uterus or womb is called pregnancy in the mother. This is the time between conception and birth. A full-term pregnancy is 28 days or 40 weeks or 9 months, but can range from 37 to 42 weeks. A total gestation is divided into 3 trimesters. A trimester is a stage of pregnancy that lasts about 3 months. The first trimester, it is from week 1 to end of 12th week. It is the most crucial and critical period in the development of the fetus. This is the period of crucial events such as implantation, differentiation of germ layers, early organogenesis, and functioning of certain organs such as heart. By the end of the first 12 weeks, all the body's major organs and structures have begun to develop. The heart is beating regularly. Fingers and toes have formed. The fetus is around 3 inches long and weighs nearly 1 ounce. The nerves and muscles work together and the fetus can make a fist. The eyelids have formed and will remain closed until around week 28 to protect the eyes. Second trimester, it is from 13th week to 26th week. It is the middle part of pregnancy when nausea might begin to settle and energy levels might increase. The fetus goes through many changes during this time, growing to be around 1 foot long and weighing 1.5 pounds. It is known as the golden or honeymoon period and is when many of the unpleasant symptoms of early pregnancy subside. The mother starts enjoying the fetal movements. By the end of the second trimester, the following will have happened. Meconium, the baby's feces, and the first bowel movement has developed in the intestines. The fetus can see, hear, make a sucking motion, and scratch itself. Skin, hair, and nails have developed. The lungs have formed but do not yet work. The fetus is sleeping and waking regularly. A male's testicles will have moved to the scrotum and a female's eggs will have formed in the ovaries. Taste buds have formed. Blood cells are developing from bone marrow. Lanugo, which is fine hair, covers the body. The third trimester spans from 27th week to the end of pregnancy, that is, delivery of the fetus or the newborn. This period is the final stage in the overall growth of the fetus and its preparation for delivery. The third trimester ends when the baby is born, which is approximately 40 weeks. This is a physically and emotionally challenging period for the mother. There is increased frequency in movements of the baby. Mother needs to nourish two lives to keep them healthy. Due to the enlargement of the uterus, the following will be difficult. It will be difficult to eat. Surrounding structures will start getting compressed. Difficulty in urination, sleeping difficulties, swollen ankles, and anxiety about the delivery. During this trimester in the fetus, most organs and body systems have formed and they will continue to grow and mature. They become capable of functioning on their own. The bones are hardening, movements become more noticeable, the eyes are open and can sense light, lung formation becomes complete, lanugo falls away and a waxy coating called vernix develops. Touch receptors are formed. Towards delivery, the fetus drops lower in the mother's abdomen and usually turns head down. The postpartum is also called as fourth trimester. The 3 months after delivery play a key role in the health of the mother and the baby. This can be an exciting time, but the range of hormonal and environmental changes can pose challenges which involve recovering overall health after delivery, especially if there are stitches. Dealing with lochia, a discharge of blood and tissue, which may continue for several weeks, which is normal. Cramping, which may feel like menstrual cramping, especially during breastfeeding. Adjusting to a new role of parenthood. Having sore breasts and other difficulties related to breastfeeding. Having fatigue due to a loss of sleep and other factors. In some cases, experiencing postpartum depression. Now, the normal delivery, what we discussed, was around 40 weeks plus or minus 7 days. Now, if the baby is delivered before 37 weeks of pregnancy, it is called preterm or premature delivery. The chances for breathing problem are more in preterm babies. The causes for preterm birth are overweight or underweight of the fetus, diabetes and high blood pressure in the mother, smoking, alcohol intake and use of certain medications by the mother during pregnancy. If the baby is born beyond 42 weeks, it is called post-mature delivery, which leads into complications for both mother and baby. Now that we have seen overview of the developmental stages, we know the fetus can develop into a normal baby or abnormal baby. And sometimes the fetus can be born dead or it can be a case of premature or post-mature delivery. Now let us see what is the fetal viability or the viability of the fetus. Viability of fetus is the ability of the fetus to survive outside the uterus or the mother's womb. The chances of viability of fetus increases if it is born after 28 weeks gestation. There are chances of more than 95% of babies to survive. However, according to the World Health Organization, fetal viability is possible after 20 weeks of fertilization or 22 weeks of amenorrhea. Medical viability is generally considered to be between 23 and 24 weeks. Babies born are often so small and fragile that they do not survive. Their lungs, heart, and brain are not ready for them to live outside the womb without intensive medical treatment. If the baby is born between 24 and 28 weeks, the survival rate is 4 to 90%, and between 21 and 24 weeks, it is 0 to 40%. As of 2022, the world record for the lowest gestational age newborn to survive is held by Curtis Zykeith, who was born on 5th July 2020 in the United States at 21 weeks and 1 day gestational age, weighing 420 grams. Now let us see what are the factors affecting viability of fetus. Birth weight. Larger babies have better survival rates. Low birth weight is independently linked to reduced odds of survival and a higher risk of disabilities and health problems. Gestational age. Premature or post-mature delivery might lead into complication. Complications. If the early birth resulted from the induction or C-section due to a medical condition such as placental abruption or oxygen deprivation before the delivery, that condition can affect the baby's health and survival. Number of babies. Singleton preterm babies are more likely to be viable than multiple babies in the womb. Oxygen deprivation. Birth complications that restrict fetal breathing, such as if the cord is wrapped around the neck, can affect survival rates. Sex. On the whole, girls have higher survival rates than boys. Steroid treatment prior to birth. Steroids taken by a mother prior to birth speed the lung development, improving the odds of survival. Steroids can help the baby to breathe normally outside the womb. And the availability of advanced medical care is very important. In low-income countries, half of newborns born at or below 32 weeks gestational age die due to a lack of medical access. In high-income countries, the vast majority of newborns born above 24 weeks gestational age will survive. Coming to the end of this session, let us understand the aim of the viability scan. Why should we do viability scan of the fetus? The aims of this scan are to ensure that the pregnancy is inside the uterus and it is not an ectopic pregnancy. This will be discussed under implantation. Demonstrate that the baby is alive. Determine the number of embryos present. Date the pregnancy and provide the estimated due date. Examine the baby for any early pregnancy abnormalities and assess the presence of internal bleeding. With this, we complete the introduction to embryology. Thank you for your patient listening. If you have not yet subscribed, please do subscribe to the channel and hit the bell notification. If you like the channel, share it with your friends and contacts. Thank you.